Hello, dear colleagues. A previously conducted molecular genetics study of a large cohort of Russian patients with congenital Aniridia revealed a typical distribution of variants in the PAX6 gene with the predominance of loss-of-function variants. But in the present study, we focused on three particular groups of variants which pathogenicity was not obvious and required an experimental investigation. The first group of our interest is splice variants. To determine the real effect of variant on the mRNA structure, we use a Minigene system. For this, we clone the genomic region containing the exon of interest with flanking intronic sequence into a special vector containing its own splicing sites. Next, we introduced the studied variant using site-directed mutagenesis, and obtained plasmids were transfected into HEK cells. After transfection, we lysed cells, isolated RNA, and performed RT-PCR analysis. Using this system, we at first analyzed 8 non-canonical splice variants found in Russian patients, 6 intronic and 2 exonic variants. To analyze these variants, we created 4 Minigene constructions and performed a Minigene assay. Our results demonstrate that 7 out of 8 variants, including missense variant and synonymous variant, disrupt normal splicing and create aberrant mRNAs. This leads to PAX6 frame shift, followed by activation of nonsense-mediated mRNA decay, and finally to PAX6 loss of function. Since our Minigene system was effective for splicing analysis, we decided to scale up our study. And now we analyze all pathogenic PAX6 variants from different databases. First, we took all PAX6 variants available at the time and performed their in silico analysis using SpliceAI tool. This allowed us to predict variants that could disrupt splicing. Analysis of variants located in introns showed that, as expected, most of them could affect splicing. Most of such variants are located in canonical nucleotides, but we were interested in those variants that are outside canonical positions and did not have a previous experimental study. Analysis of variants located in exons showed that approximately 15% of them could affect splicing. Such group includes synonymous, missense, and even nonsense variants. Out of them, for further analysis, we took all synonymous, all missense, and part of nonsense variants. In order to experimentally analyze interesting variants, we improved our Minigene expression system and created several new Minigene constructions. So now we could analyze splicing of all PAX6 coding exons. In total, we analyzed 38 intronic and exonic variants and showed that most of them actually disrupt splicing. It allows us to establish the real molecular mechanism of the pathogenicity of studied variants, which is especially important for exonic variants. The second interesting group of PAX6 variants are variants located in 5' untranslated region. We found 4 such variants in Russian patients. Additional 14 variants were also reported in the literature, but mechanism of their pathogenicity was unclear. We performed a functional study for 5 such variants. For this, we cloned a full-length PAX6 5' UTR, including full-sized introns, into luciferase vector and fused 5' UTR with luciferase. Using site-directed mutagenesis, we introduced studied variants, and all obtained plasmids were transfected into 2 cell lines. After this, we performed RT-PCR, qPCR, and luciferase analysis. RT-PCR of transfected cells showed that 2 intronic variants led to an exon 3 skipping, while 3 exonic variants did not affect splicing. Analysis of protein and mRNA expression showed that all variants lead to a depletion of protein level, while mRNA expression level was not significantly reduced. So we conclude that studied variants decrease the protein translation efficiency. To understand the underlying mechanism of how variants influence on translation efficiency, we performed a bioinformatic analysis as well as experimental validation, and we showed that there is a small upstream open reading frame in the PAX6 5' UTR. Moreover, we revealed that all studied variants disrupt this upstream ORF in the same manner. Splice variants, as well as small insertions and deletion, lead to the same upstream ORF frame shift and formation of extended ORF. This extended ORF overlaps with downstream PAX6 coding sequence and thereby reduce its translation efficiency. Thus, we discover a new molecular mechanism of congenital Aniridia pathogenesis and showed that this mechanism is the main one for almost all known PAX6 5' UTR variants. To sum up my story, I want to highlight that in our laboratory, we develop and successfully use systems for functional analysis of different PAX6 variants. At the moment, we have a well-functioning system for analysis of splicing variants located in the coding part of the PAX6 gene. Besides, we have a luciferase system that allows us to analyze variants in PAX6 5' UTR, their effect on splicing, mRNA, or protein expression. And in addition, in the present time, we are developing a system that will allow us to analyze missense variants in the PAX6 gene. Using the luciferase system, we will be able to evaluate the efficiency of PAX6 protein binding to its targets. So now we are open to collaboration, and we will be happy to analyze other interesting variants in the PAX6 gene. If you are interested, please contact me. And in the end, I want to thank my colleagues who participated in this work, and thank you for your attention.
We don't have any way to make questions, so I think that the session is closed. I want to thank, on behalf of me and Juliana Martinez, my co-chairperson, your kindness to stay here so late with this weather, you know. So I think that is a perfect moment to go for a walk, to go for dinner, and thank you very much for your kind attention to this session and to this today, to the meeting. Thank you very much, and the session is closed.
Bye.